Hi everyone, Anna is here, and in this video we're going to discuss the topic of B lymphocytes. So if you are not sure about their structure and how they work, then this video is for you. Compared to phagocytes, B lymphocytes constitute primary specific immunity, meaning that B lymphocytes will be acting against particular antigen or pathogen. So I'm going to divide this video into two stages. So what happens to B lymphocytes before they encounter pathogen and then after. The lymphocyte structure has a large nucleus and they're much smaller than phagocytes. You might be asked to draw this sometimes in exams, so do please remember this. B lymphocytes are produced in the bone marrow as many other white blood cells. And this is the place where they also undergo maturation. Maturation means they will be selected against a specific antigen. So there'll be some gene expression going on that will make them specific and in complementary to binding to specific pathogens. And so when they do mature, the B lymphocytes look like this. So it's a lymphocyte and there are antibody receptors that are attached to the cell surface membrane. Once B lymphocytes are matured, they're released into the blood and they will be circulating around the body, and they'll particularly concentrate in the liver and spleen. So these steps summarize everything that happens to B lymphocytes before they encounter pathogens. Now let's go and look at what happens when they do actually encounter a specific pathogen. The B lymphocytes can encounter pathogens in two ways, either directly in the blood circulation or by the antigens which are brought by the antigen-presenting cells such as macrophages. So once the pathogen, or let's just keep referring to it as an antigen, will be brought to the specific beta lymphocyte, this will result in the complementary binding between the pathogen/antigen to the B lymphocyte. And this, in turn, will result in a process that is called clonal selection. Clonal selection specifies that this B lymphocyte is complementary to this specific antigen, and so therefore the immune system will now go and expand these populations of B lymphocytes in the process called clonal expansion. And clonal expansion includes the process of mitosis. So the cells will produce the clones of that B lymphocytes, and this will result in a huge population to combat the pathogen. After the clonal expansion, the B lymphocytes will take two routes. They will differentiate in two different populations. One is going to be the population of the plasma cells, and the second population is going to be the population of the memory cells. Plasma cells do not have antibody receptors bound to the cell surface membrane, and instead they're the cells that produce antibodies. And in my next video, I'm going to discuss the structure of the antibodies and their downstream functions. So plasma cells produce antibodies, and then the second population which B lymphocytes differentiate to are the memory cells. And the memory cells, they're important for the secondary immunity. So when the same pathogen enters the organism second time, then the memory cells they will differentiate back into plasma cells, and they will result in a much faster production of antibodies. This is it for B lymphocytes topic. In the next video, I'm going to talk about the T lymphocytes and antibodies. So please subscribe to my channel. Thank you so much for your likes. Please do support the channel, and I will see you in my next video. Goodbye.